Unlocking the secrets of fat tissue, the inflammatory response in obesity. New single-cell study uncovers diverse immune cells in obese mice. Fat tissue, for as much as it's been vilified, is an incredibly complex and essential bodily organ involved in energy storage and hormone production, among other functions. Yet, modern lifestyles have led to a worldwide epidemic of obesity and a corresponding increase in related conditions like type 2 diabetes and cardiovascular disease. Researchers are attempting to uncover the basics of how fat tissue is structured and specifically inflammation associated with obesity in the hopes of unlocking the connection between the accumulation of fat and poor health outcomes. A cutting-edge study on fat tissue. A new study from Lindsay Muir, PhD candidate Cooper Stansberry, and their colleagues uses single-cell analysis of gene expression combined with spatial transcriptomics to reveal previous unrecognized immune cell types and interactions within adipose tissue. Spatial transcriptomics is a newer technology that captures all gene expression in small spots across an entire thin section of tissue. Studying fat is easier said than done. In tissues that are organized into defined layers, for example, the spinal cord or the brain, it's easier to do sanity checks with your data and identify this or that layer as a particular cell type and know that it should be expressing genes X, Y, and Z, said Muir, a research assistant professor in the Department of Computational Medicine and Bioinformatics. With adipose, it's a lot more challenging because the cell types are distributed evenly throughout the tissue, without defined cell layers. In obesity, fat cells, or adipocytes, expand and can reach a limit that ultimately causes cell death and leads to inflammation. The experiment, high-fat diet in mice. To better understand the types of immune cells within adipose tissue and where they are in relation to each other in obesity, the team fed mice a high-fat diet over the course of 14 weeks, collected fat tissue, and then used single-cell and spatial analyses to produce a readout of all the mRNAs present in the sample. Using a computational process known as clustering in the single-cell data, they were able to group cells whose genetic makeup more closely resembled each other than other groups or the overall sample. They discovered something surprising about the sample's population of macrophages, an immune cell whose job it is to clean up dead cells and debris. We knew going in that macrophages would likely have multiple subtypes. What surprised us were the number that came out that were highly different from each other, and coming up at different times and becoming more dominant over time. They identified 5 types, which they named MAC 1, 2, 3, 4, and 5. MAC 1 was resident to the tissue in both lean mice on a normal diet and obese mice. MAC 2 and MAC 3, which were identified by their pro-inflammatory genes, peaked after 8 weeks of the high-fat diet. However, as the high-fat diet progressed to 14 weeks, MAC 4 and MAC 5 cells, which had low pro-inflammatory gene expression, predominated, while pro-inflammatory MAC 2 and MAC 3 cells decreased. The thinking in the field has been that the type of macrophages that accumulate in obesity are promoting an inflammatory state. Based on these data, there's a lot more to the story, said Muir. Her hypothesis is that MAC 4 and MAC 5 are the lipid-associated macrophages, LAMs, described in her own prior work and by other researchers, and may be a sign of the body attempting to quell a damaging level of inflammation from pro-inflammatory macrophages and dying adipocytes.